Hello, my name is Tom Stricker, and I really appreciate the opportunity to speak to you today, and I appreciate the invitation from Simon. I wish I could have been there in person, but we weren't able to work out the travel arrangements in time. So we're going to do this from a recording, and hopefully it'll work. We're going to start actually with a bit about the molecular test we are currently developing for sequencing cancer at Vanderbilt, and then we're going to talk about where some future plans, being able to take that test to liquid biopsies and assaying circulating DNA. So this is data that was collected from Vanderbilt by William Powell and Mia Levy that discusses our experience with snapshot in lung cancer. So snapshot, we were measuring point mutations in a handful of genes, including EGFR, BRAF, AKT, PTEN, PIK3CA, NRAS, KRAS, etc. And there's a couple important points to take away from this. One is that there's a number of mutations that occur at pretty small frequencies, so 2% for PIK3CA, ROS1 fusion, RET fusion at about 1%, but that these mutations, even though that they're low frequency, can have important impacts on therapy, as there are targeted therapies for these fusions or mutations. And so testing all of these sequentially, it becomes not only a technical issue, but honestly, with small biopsies and such, we can run out of DNA. And so having it, and then testing for low frequency mutations one at a time is also pretty inefficient. If you have to test 100 cases with a ROS1 for a ROS1 fusion just to get one positive, that's not terribly efficient. But if you had a test that had all of these, or the ability to detect all of these mutations on a single test, we wouldn't run out of DNA. And the other upside to that is we have this large gray box here, which is there's no mutation detected. Presumably that's because in our snapshot assay we're not effectively assaying for the correct genes. And at Vanderbilt, we're working on a platform that is a single platform that will detect 4 mutation types. We call them the 4 horsemen: point mutations, obviously, and indels, copy number changes, amplifications, and deletions, and then translocations, which are the trickiest. And for right now, we're focusing on a narrower set of those. So once you've decided that you're going to sort of target 4 different mutations, so once you've decided to build a test that can detect the 4 horsemen, you're faced with another issue, which is that in many of our tissue sections from patients, many of the nuclei will be normal and will be diluting out the tumor DNA that's present in our sample. And so in the upper left corner here, we have some islands of epithelial cells. Those are the pancreatic tumor. All the dark blue dots on the middle and sort of lower left corner, those are lymphocytes. Each one of those dots is a nuclei. So there's actually a lot more DNA that's going to come from normal lymphocytes than are going to come from the cancerous pancreatic glands. And this is a real issue. So there's 2 ways to fix it. One is macrodissection, which on a slide. So this is data from Foundation Medicine's paper and Nature Biotechnology, but you can see this is a mixing experiment where they mixed known samples at low, greater than 10%, between 5 and 10%, and less than 10% allele frequency, and then measured their ability to detect them at different coverage levels. And so if you sequence deep enough to say 600 or 700, you can so you're covering your average coverage for every base that you are targeting is around 700. You can get to pretty good detection sensitivity for even low allele frequencies or high normal contamination within your tumor. All right, so we have another issue, which is that the human genome is a very complicated place. It's about 3 billion base pairs, and roughly 1% of that is coding. And for the most part, we're really only interested in coding sequence, mostly because that's all we can interpret at this point. And certainly mutations and regulatory elements of promoters and enhancers even will probably prove to be interesting, but just in terms of actionable, clinically actionable variants that we can interpret, we're really focused on the coding sequence. So we'd like to sort of focus our abilities or focus our sequencing on just the pieces of DNA in which we're interested in. So there's a couple different ways to approach target enrichment. One's an amplicon-based approach. The pros here that it's quick and efficient, and it requires very low sample amounts. You can even push down to say 10 nanograms. This is good for small gene panels because the number of genes in a panel is limited. You're running multiplexed PCR in a single tube. So clashes between primers and efficiency between primers, all that sort of stuff can be hard to control. You typically do get uneven PCR, which then equals uneven coverage, and so the coverage bounces around quite a bit. It's possible to get copy number information. Translocations are very difficult. But the copy number is actually pretty hard as well, and you have to make sure that you're not over amplifying, etc., and of course, variants outside amplicons are not sampled. A second approach would be target enrichment for using hybrid capture. So this is how most exomes are done. Very briefly, you start with the whole genome. You break it up into tiny pieces. The light blue here are the pieces of DNA we're interested in. The dark blue are the pieces of DNA we are not interested in. You have a bait set that's usually linked to biotin. That bait set is complementary to the pieces of DNA for which we are interested in, so those bind to our baits. We then use streptavidin to bind those baits to magnetic beads, and we can wash away the pieces of DNA we're not interested in. The pros here is that you can have a very large capture space, up to and including the entire exome. Copy numbers and translocations are more tractable, although I wouldn't say that they're easy. And variants outside the baits might be sampled, just because this washing away part isn't, well, for two reasons. One, the washing away part isn't perfect, and so random pieces of DNA come down. And two, because you tend to get almost like a normal distribution around the baits, and so because of the way the libraries are built, it's called splash, and you sequence a little bit. You tend to get some sequence on either side of where the probe actually sits. The cons are it does require a larger amount of DNA. Certainly makes FNAs or even some small needle biopsies hard to approach with this technology. The sequencing is relatively inefficient, although it's certainly getting better. Because of this splash idea, some of the bases you're generating are off target because we don't perfectly wash away all the junk DNA that we're not interested in. Some of the sequencing is off target, and the more sequencing that's off target, the more you have to sequence to get the bases you need to be on target. And the library builds are slow. Typically the HYBs, most of the commercial HYBs are anywhere between 24 to 72 hours. People are pushing those lower, and there certainly have been a lot of improvements in this technology, but it does, right now I would say it's still definitely slower than standard library build, or amplicon-based build. So how do we detect fusions with hybrid capture? So this is actually data from William Powell. So he had helped us get this set up before he left for Roche. But essentially, we pick genes of interest, so kinase genes, JAK, ABL, ROS, NTRK, BRAF, PGFR beta, ALK, RET. We identify where the kinases, the exons that carry the code for the kinase domain, because all of the translocations are going to happen upstream of the kinase domain, because we're going to maintain that kinase domain if it's going to be functional. And then so we just tile probes through those regions, avoiding repeats, and then hope that when we catch a fusion, that essentially we have reads mapping. We have reads that are pulled down and that map to where paired end reads, where one end of the molecule maps to our kinase and the other maps to whatever the fusion partner is. And that was, so V1 of our panel used these this strategy to target 19 fusions for 19 genes, mostly kinases, but also temprosets too, and a couple recurrently translocated transcription factors. We're actually switching to. Now, I don't have a slide to show it, but we also have probes that are tiling throughout the genome. We have one about every 5 million base pairs or so. These probes are targeting, so there's about 4,000 of them. These probes are targeting common polymorphisms. And that's what we're going to use. We can use those to build a coverage model across the entire genome, and then basically segment that coverage model to identify regions of amplification and deletion. And so that's how we intend to identify those. So standard sequencing for of hybrid capture to identify point mutations and indels, anchored PCR of RNA to identify fusions, and then tiled probes targeting common polymorphisms to identify copy number changes. And then, so our first design, we tested on HapMap cell lines and some cancer cell lines, had some very good results. And then rather than bring the sequencers into the CLIA lab, we have started a collaboration with a company, BioReference Labs Incorporated, to help us bring our panel into the NIC. And that led to a bit of a change in our panel. We're able to use some of their workflows in order to, I think, even improve our V1 panel even more. So we settled, I think, on 297 genes. We've designated about 100 genes of that as core content, and those are done on an amplicon-based approach. And then we have the remaining genes plus the copy number probes are built as a hybrid capture approach, and then we have the anchored amplicons out of RNA for translocations. And this modular design allows us to do a few different things. So if we have a very small amount of DNA, or we have a small sample that we know is going to produce a small amount of DNA, a core needle biopsy, we can at least get the amplicon-based approach to work because we don't need much DNA there. And plus the anchored amplicon-based approach will also work. So we'll be able to get information on really what are probably the key genes, anyways, for those patients. And then as this content's unlikely to change, but these 200 genes might change more often. So as we have to go through regulatory, it's a little bit easier to swap this out without causing too much harm. And then if we want to switch to, we want to increase our fusion list for sarcomas, for hematologic malignancy, etc. Again, we're just changing this module. We're not live yet. We hope to go live this summer, and I think it'll be interesting to see how ordering patterns are affected, and whether we're just going to also offer the 100 gene core content as a sort of standalone, or the fusion standalone, and see how physicians order and use this test. All right, now we're going to transition a bit to where, once we've got the next gen panel up and running, where we're going to push that next. And so we're really interested in the liquid biopsy. There's been a lot of work on this from lots of people. So there are short fragments of DNA floating around in everyone's blood. There's some idea that it might be enriched for nucleosomes, so short fragments 1 to 200 base pairs. If you remember, there's 147 base pairs wrapped around the histone core, so right in the right size for nucleosomes. The protein perhaps is protective of the DNA. There's very small amounts of this, something like 500 to 1,000 genome copies per mil, so it's not plentiful by any means. And there's been a couple different approaches to sort of assaying this DNA, both whole genome approaches as well as targeted approaches. And the targeted approaches include both sequencing and non-sequencing approaches. You can do this basically. There are a number of clinical situations in which it has been suggested that liquid tumor biopsies could be beneficial for the patient. Many of these are when there is a second genome that is circulating or is present in the patient. So one obvious example is organ transplant. Detection of the donor genome or increases of the amount of the detectable donor genome in the blood could correlate with rejection or other damage to the organ that's ongoing. And maybe we could use this instead of frequent needle biopsies of the organ as sort of a first line to decide whether we needed to biopsy the organ or not. Another instance where we have a second genome is obviously pregnancy, and it's been people have suggested the use of sequencing circulating DNA to detect aneuploidies or other point mutations within the fetus. This could replace amniocentesis, at least as a screening tool, and reduce the number of amniocenteses that are done, and thus the risk for miscarriage that comes with them. And finding malignancy is really a sort of instance where we have two genomes or more within the individual, and being able to detect mutations in the blood could be used as a screening device. It could be used as a therapy monitoring test, and it could be used perhaps to detect resistance mutations as they develop to targeted therapies. So we're talking about a couple different techniques. This is from Bert Vogelstein's lab. So the idea here again is that we have a tumor that's growing in the body. It's contributing DNA to the circulating cell-free DNA and plasma that can be isolated and then sequenced, and then we can use that to identify translocations, amplifications, deletions, chromosomal gains and losses. So in this study, they built whole genome libraries, generated about half a lane of Illumina data, something like 100 to 200 million reads, which is insufficient to cover a full genome. But then essentially what they did is mapped those reads to the genome and then just counted the chromosomes. So if you had more reads than you expect by chromosome length, that was a suggestion of aneuploidy. And this is actually the technique that is obviously used for fetal aneuploidy as well. This is So they have several normal controls where they see very tight chromosomal counts, and then several cancers where they see a wide distribution of chromosomal counts indicating aneuploidy. And this works because every read is a count, and so even though you're not sequencing very deeply for a genome and the tumor isn't present, you're generating millions of counts per chromosome. And so your accuracy in measuring the amount of each chromosome in the plasma is pretty high. So it's actually quite sensitive. And I just wanted to note, like this reagent cost is something like $750. So one does wonder if this could replace screening tests or estimates, or be used to estimate tumor burden within patients. Now, what it doesn't really tell you is anything actionable. It does give you some sort of crude measurement of the amount of tumor circulating in the blood, but it doesn't really tell you anything about which mutations a tumor might have, if there are any therapeutic implications, etc. Of course, if we've performed a sequencing panel on a patient prior to wanting to monitoring them for circulating tumor DNA, we actually know where the mutations are, genotyped them, and so we know there's, like, for instance, a mutation here for this patient, so we can build an amplicon-based sequencing approach that would specifically look for these or whatever other mutations they had in their tumor to see if to monitor disease levels, response to therapy, etc. This would be a targeted approach, and there's a couple different possibilities. So one would be using digital PCR with essentially probes or primers directly targeting the mutant allele, and the other would be NGS, or next generation sequencing. One of the issues with next generation sequencing, of course, is that its error rate, especially on the Illumina, is higher than what we think the detectable allele frequency for a tumor might be, where you have an allele frequency of, say, less than 2% and an error rate of more than, or right around 2%. So in order to use next generation sequencing in this manner, we're going to have to modify the protocol somewhat to try and improve that error and increase our sensitivity so that we can detect very low allele frequencies in circulating tumor DNA. One way to do this would be essentially this mechanism here where we barcode primers, so that there's essentially like a randomer in each of our universal adapters. That way, errors that are generated during PCR would not be considered a low frequency mutation, because not all of the randomers here have the mutation, whereas true mutations would be present. We would have more than one copy with the randomers, the same randomer present. Digital PCR is reasonably straightforward. Here we take whatever we want, genomic DNA, cDNA, RNA, etc., whatever your favorite plasma. That is distributed into, or partitioned into many small reactions, 20,000 or more, where each well either gets 1 or 0 molecules of nucleic acid, and then we have a detection system that detects positive and negative reactions. This gives us, we know the distribution for which you can have positive and negative droplets, because it's going to be based on the Poisson distribution. And so this enables us to calculate absolute concentrations of target molecules, and is extremely sensitive for this. And so it allows us, you could build assays that specifically detect mutant alleles for patients for which we know the mutations are present in their primary tumor, and then assay their blood repeatedly, looking for increases or decreases or changes in the amount of these mutations that are present in their plasma. Working with Simon to bring automation and liquid handling into VANTAGE, which is our sequencing core at Vanderbilt, using the amplicon portion of the panel that we developed, but being able then to have essentially a plate that represents that amplicon portion of our large panel, being able to cherry pick individual portions, amplicons, to build a custom pipeline, a custom panel for individuals, and then adding in known resistance markers, and then building a essentially personalized circulating tumor DNA assays from CAP and CLIA certified primer sets. So to reduce both search space and cost, and hopefully allow us to build these personalized panels to be able to monitor disease and resistance in individuals. So in conclusion, advances in sequencing technologies will allow us to cheaply, quickly, and sensitively sample circulating DNA. These assays may allow us to replace or reduce the use of more invasive assays. And of course, challenges abound and include both regulatory infrastructure, reimbursement, and ethical issues. I would love to be able to be there in person so that we could have discussed these issues, but I think the plan is for me to try and call in now on a GoToMeeting. Hopefully that's going to work. If it doesn't, please, if you have any questions, I'm sure Simon can direct you to my email. I'd like to acknowledge Mary Zuter, who I worked very closely with in developing the cancer panel, Sam Santoro, who is the chair of pathology, Jennifer Pietenpaul, who's the director of the Cancer Center at Vanderbilt, Kim Dahlman, who runs a translational core lab, which I worked really closely with, again, in development of the panel. And then a lot of oncologists, William Powell, Carlos Arteaga, and Christine Lovely were particularly involved in developing our panel, but a lot of members of the VICC took a look at it and made sure that we were covering the genes they were interested in. The staff at VANTAGE, which is our sequencing core, have been super helpful, and then Robert Daber and Chirag Desai at Bioreference Labs have been wonderful to work with, as well as we work on bringing this panel to the clinic. Thank you all for your time.